The following video will provide an overview of the kinase specificity database. To access the kinase specificity database, click on the link in the pLogo navigation bar at the upper right. The purpose of the kinase specificity database is to provide interactive pLogos to represent the specificities of kinases in various organisms. The complete list of kinases is sorted in alphabetical order by protein name at the bottom of the page. To browse the list, click on the arrows at the right and left, or click on the numbers at the bottom to jump to a particular page. Following each kinase name is a number and a letter. The number represents the number of unique substrate phosphorylation sites for that kinase, and the letter represents the residue on which they are phosphorylated, in this case serine. Above the kinase list, various options exist for sorting, viewing, and filtering data. At the top is the organism filter. At present, human, mouse, and rat phosphorylation data is available. Below the organism filter is the database filter. By default, Phosphocyte Plus data is selected. You can also add data from the phospho-ELM database. Below the database is the residue. By default, serine phosphorylation sites are shown, however, threonine and tyrosine can also be added. Below the residue is the selection mode. In single selection mode, clicking on a kinase will provide a pLogo for that particular kinase. In multiple selection mode, the pLogo for multiple kinases can be merged. Below the selection mode is the sort by option. You can either sort by kinase name, as is default, or sort by the number of sequences available. Finally, you can filter by name. If you are looking for a particular kinase, you can filter for that kinase by typing it in here. Let's say we were looking for the kinase CDK2. And we wanted to find serine as well as threonine phosphorylation sites. Here we can see 4 entries result, 2 from the Phosphocyte Plus database and 2 from the phospho-ELM database. To see the results from the Phosphocyte Plus database for serine, simply click on that entry. The pLogo then appears in the pLogo window. Hovering over the pLogo shows statistical information about each residue at each position. You can also click on a particular position to fix that position to recalculate conditional probabilities. To merge the results from both the Phosphocyte Plus and phospho-ELM database, you can enter the multiple selection mode. In this mode, selected kinases will appear in a kinase list in the kinase box at the upper right. Let's click on CDK2 from phosphocyte, as well as CDK2 from phospho-ELM, both serine-centered. Clicking on the Generate pLogo will create the hybrid pLogo that results from merging these two databases. We can also add threonine data and regenerate the pLogo that way as well. Clicking Clear List will clear the list and clear the filter by name by deleting the characters in the text field. Note that at least 5 substrates are required to generate a pLogo. Thus, if we select a kinase like CDK11 with only one known substrate and try to generate a pLogo, an error message appears notifying us that the minimum number of sites for pLogo generation is 5.